age where science reigns supreme and every phenomenon is explained through laws, formulas, or mathematical models, there remains one field that silently humbles researchers: DNA, the miraculous genetic code within every living cell. Each of us, our appearance, personality, even our diseases or genius, is written in a language no one can see, yet every one of us carries within. And that language, many scientists believe, is not merely the product of random mutation or natural selection. So what if DNA isn't the result of randomness, but of design? If the genetic code is truly a message sent by a supreme intelligence, by God or a cosmic will, then what is being conveyed? And what if modern scientific experiments are gradually lifting the veil between science and spirituality? Two realms that have seemed opposed for thousands of years. What exactly is DNA, and who, or what, could perform the magnificent feat of compressing matter into something that holds such vast amounts of information? And why do some scientists say they have seen God when studying DNA? Today, let us step into the microscopic world, the world deep within each of us. DNA. Francis Collins. He is the director of the U.S. National Institutes of Health, formerly the director of the National Human Genome Research Institute, and also the leader of the famous Human Genome Project. Most people have at least heard of the term gene. So what exactly is the Human Genome Project? Let's take a closer look. Human cells contain 23 pairs of chromosomes that carry genetic information. This includes 22 pairs of autosomes and 1 pair of sex chromosomes. These 23 pairs of chromosomes are inherited equally, half from the father and half from the mother. These chromosomes contain a total of 6 billion DNA base pairs. The mission of the Human Genome Project was to determine the precise sequence of these DNA codes, provide a comprehensive genetic family portrait, and identify the specific gene segments responsible for genetic functions. The ultimate goal of this project was to decode human genetic information. Later, they discovered that the more they studied, the more complex it became. American researchers began to realize that they might not be able to handle it alone, so they began expanding international collaboration, seeking support from other countries. In this way, countries like the UK, Germany, France, and Japan gradually joined the project. Eventually, the Human Genome Project became a massive, interdisciplinary, and multinational scientific endeavor. To date, humans have completed the sequencing of 92% of the genome. The remaining 8% of the genome remains unsequenced for now due to the current limitations of technological capabilities. This double helix structure of DNA was discovered in 1953 by American biologist James Watson and British biologist Francis Crick. When this discovery was unveiled, it completely shocked the scientific community. And from that moment on, humanity opened a great door to explore the secrets of heredity. The transmission of genetic information in DNA is composed of 4 nucleobases: A, adenine, T, thymine, G, guanine, and C, cytosine. From here on, we will use the 4 letters A, T, G, C to represent them. These 4 nucleobases pair up to form 2 base pairs. AT is one pair, and CG is the other. With these base pairs, different combinations determine the physical genetic traits of an organism. For example, height, body size, and even non-physical traits such as personality and intelligence. On each chromosome strand of a living being, there is a long DNA molecule wrapped around histones. And this DNA molecule is presented in a double helix structure. It coils inside the cell nucleus, which is only about 6 micrometers in size, seemingly very small. However, if we were to stretch it out, the DNA could reach up to 2 meters long, taller than the average adult male height globally. If we use the four letters A, T, C, G to represent the double helix structure of DNA, doesn't it resemble the punched paper tape used in early computer programming? On the paper tape, a punched hole represented a 1, while an unpunched spot represented a 0. In the programming world, binary code is used, and regardless of which programming language a coder uses, in the end, it all has to be converted into machine language composed of combinations of zeros and ones. So in the words of Dr. Collins, if we apply computer programming logic to the world of DNA, we can say that DNA uses a quaternary code, that is, the four codes A, T, C, G. The amount of information contained in this quaternary code is far greater than that in a binary system. For example, if we have 20 code units, using a binary system would yield 2 to the power of 20 combinations. That is, 1,048,576 combinations. But if we use a quaternary system, the number of possible combinations would be 4 to the power of 20. This number is roughly 1,000,000 times larger than the binary one. In other words, the more code units there are, the greater the difference becomes. So, how much information is actually stored in human DNA? Let's try to calculate it. In a human body cell, there are 23 pairs of chromosomes with a total of 6 billion DNA base pairs. And one important conclusion drawn from the Human Genome Project is that the autosomes of males and females are the same. There is no difference. From this, we can say that human genetic information is made up of 3 billion quaternary code units. In other words, the amount of information that can be stored in a tiny human cell is 4 to the power of 3 billion. Such a tiny space capable of storing such an enormous amount of information. This is truly the ultimate dream of computer storage systems, and just a combination of one or a few specific codes among these 3 billion DNA bases can control the human body and perform various complex functions. Human life begins from a tiny fertilized egg, which continuously replicates and divides, replicates and divides. Eventually, the entire body composed of all cells will have identical DNA in the chromosomes within each cell nucleus. Yet once these cells enter the process of division, the cytoplasm can undergo endless transformations, forming various types of cells and tissues in the body. For instance, red blood cells, white blood cells, nerve cells. Humans also develop different organs. Moreover, biologists have discovered that during the developmental stages of a human embryo, different structures and organs emerge at different time periods. Even these specific time points have been predetermined. If development happens too early or too late, the child will be born with congenital deformities, and all of this is precisely controlled by DNA. According to conventional thinking, top scientists in life sciences, like Dr. Collins, are thought to be people who, with just a slight tweak or pull, can alter the genetic code of life, even create entirely new hosts. However, this is difficult to achieve due to the enormous risks involved. One highly controversial case involved He Jiankui, an associate professor in the Department of Biology at the Southern University of Science and Technology in China, and his research team. They conducted genetic editing on human embryos, which sparked a massive outcry. At the time, they aimed to make children immune to the HIV virus. The incident shocked society as a whole and received widespread condemnation. Beyond the ethical concerns, the technical issues and potential future hazards were all reasons why the scientific community strongly rejected this endeavor. Based on current human understanding of DNA, altering human genes at will is highly questionable. Who can say whether the gene-editing scissors are cutting out the potential for AIDS or cutting into the future of humanity? In gene-editing experiments, there is a known risk called the off-target effect, meaning that during the gene-editing process, other unintended genes might be inadvertently damaged. As a result, this can lead to outcomes such as gene mutations, genetic defects, or chromosomal translocations. Let's take software programming as an example. Imagine a massive source code, a superprogram with immensely powerful functions. People only know what outcomes will appear once the program runs, but they can't see the source code clearly. At most, they might understand just a small part of it and wouldn't dare to casually modify the code, fearing that certain critical segments are deeply entangled with others. Touching one line could break the whole system. In the same way, through the 4 codes A, T, C, and G, 3 billion sequences of DNA are written, creating the human body with its perfect structure, advanced intelligence, and stable biological state. Can this truly be just the result of a series of coincidences and random interactions? Moreover, genes possess a powerful replication function. When one cell divides into 2, its DNA rapidly replicates so that both cells carry identical DNA. This process happens extremely quickly and continuously. At every moment, countless replications are taking place in our bodies. If supercomputers are created by intelligent life, then who created the functions of DNA? DNA possesses a powerful self-repairing system. Scientists have discovered that when DNA is damaged or when errors occur during replication, it has the ability to repair itself. This includes position repair, excision repair, and reorganization repair. As of now, more than 140 different DNA repair mechanisms have been identified. Even if a repair becomes impossible, the DNA's recovery system can still activate a self-destruction mechanism, so that the faulty genetic information doesn't spread further. The cell will vanish along with its damaged DNA. In truth, even the best software engineers today cannot write a program as intelligent as this. With its incredible strength, precise replication, and self-repair mechanisms, DNA ensures an extraordinary level of stability for biological genetic information. The Human Genome Project found that from the moment humans first appeared until now, after being passed down through tens of thousands of generations, people across the globe, regardless of region or ethnicity, have DNA differences of only about 1 in 1,000. And this 1,000th difference accounts for the individual characteristics and traits of you, me, and everyone else. It does not mean the emergence of a new master species, yet the wonder and mystery of DNA doesn't end there. Thousands of years ago, long before electron microscopes existed, and before modern science had even laid its foundations, our ancestors across various civilizations carved and painted mysterious symbols, from the interlocking circles in Indian mandalas, to the mystical spirals in ancient Egyptian art, to the flower of life etched into stone at Abydos. It seems they were trying to pass down something that transcended time. Strangely enough, these seemingly abstract geometric patterns, when compared to modern science, reflect the precise structures of fundamental components within the human body, especially DNA. The double helix structure of DNA is not a random design. Upon closer mathematical inspection, one finds that the ratio between the twist cycle and its length, about 34 angstroms per twist to 21 angstroms in diameter between the steps, forms a 34:21 ratio. Dividing 34 by 21 gives approximately 1.618, the golden ratio, phi. This ratio is often considered the code of beauty and the order of the universe found in art, nature, and even sacred ancient architecture. Each twist of the DNA forms a symmetrical helix band, as if it were designed with aesthetic logic rather than randomness. Many mystical biologists have suggested that DNA is a living mandala, a sacred symbol reflecting the balance of the universe in every living cell. Recent experiments from Russia and Germany have shown that when specific frequencies of sound waves or light waves are directed at DNA, the genetic sequence can actually change. But not just any frequency works. Only those that follow harmonically structured waveforms, like golden waves, spiral waves, or mandalas, can influence genes. Some researchers have even asked, could prayer, meditative music, or chanting, each following sacred harmonic tones, be genuinely interacting with our DNA? Could it be that faith and sacred intention do more than comfort the mind? They might actually reprogram our biology. When we observe the same geometric principles present in the structure of seashells, the spiral arms of galaxies, and even in DNA, we cannot help but ask, who designed this system? Some modern quantum physicists propose that the universe is a spiraling information matrix, and that sacred geometry is the fundamental language used to store and transmit knowledge across space and time. In other words, DNA, proteins, cells, are not merely biological. They may be encoded data from a higher intelligence, what humanity has long called God. And now, looking back at the DNA genetic code, so complex, yet so powerful, perhaps you can begin to understand why leading scientists like Dr. Francis Collins have found that the deeper they dive into research, the more they see God everywhere, right? Dr. Collins once shared that he was truly an atheist. But that was in the past. Ever since entering the field of medical research on DNA, he began to feel a much deeper connection to the essence of life. In an article Dr. Collins wrote for CNN magazine in 2007 titled, "Why This Scientist Believes in God," he stated, "I see DNA, the information molecule of all living things, as God's language. The elegance and complexity of our bodies, and the rest of nature, is a reflection of God's creative spirit." As humanity continues to explore DNA more deeply, many, just like Dr. Collins, begin to sense the presence of something greater. Could DNA be a sacred code, seeded into the universe from the very beginning? And if that's true, perhaps it's time we take a moment to reflect that within every cell of our bodies lies an entire universe, written in the language of God. What did you think of today's video? Share your perspective on this topic in the comments below, and don't forget to like, share, and subscribe to support the channel so we can continue making more valuable content for you. Thank you so much.